Students, welcome to Biotech Academy. Today we'll talk about protein denaturation. So, what are proteins? We've already discussed. These are the most important class of biomolecules which are involved in regulating and carrying out essentially all the reactions that occur inside the living system. And these play very crucial role within the living system because they are the key molecules which regulate all the metabolic reactions that occur for the cells for the living system to sustain. So these are one of the most important biomolecules, and we've also seen various levels a protein folds into to attain the functional molecule. So we've already discussed in our previous lecture there are 4 levels of protein structure: primary, secondary, tertiary and quaternary. And these structures, the attainment of these tertiary and quaternary structure is essential for the protein to attain its function, and correct folding of the polypeptide chain, linear polypeptide chain, into correct 3-dimensional structure is essential for the protein to perform its function. And to attain that 3-dimensional conformation, there are various factors or various type of interactions which play pivotal role. So various interactions include covalent and non-covalent interactions. We've already discussed all about each type of interactions in detail. So covalent interactions include disulfide bridges, and non-covalent interactions include hydrogen bonds, ionic bonds, van der Waals forces and hydrophobic interactions. And it is a cumulative effect of all these covalent and non-covalent interactions which imparts the correct 3-dimensional conformation to a linear polypeptide chain, which is composed of amino acid residues linked together by peptide bonds. Okay, so what is denaturation? Denaturation is the process where the protein loses its native conformation. Native conformation is the 3-dimensional conformation in which the polypeptide chain folds into for its performing its function or for carrying out the correct function. And it is the cumulative effect. It is the summation of all the favorable interactions in the form of non-covalent and covalent interactions which outweighs the sum of unfavorable interactions which leads to the correct folding of the protein. So in the process of denaturation is when these interactions they break and the native conformation of the native 3-dimensional conformation of the protein is lost. Okay, so what is the how this denaturation process occurs? It may occur because of changes in the surrounding conditions which causes the breakage of these covalent or non-covalent interactions. Remember, when we were discussing about the covalent and non-covalent interactions, I already mentioned there are certain strong interactions whereas certain weak interactions. So weak interactions, even slight modification in the surrounding conditions leads to the breakage of those interactions. So for example, ionic bonds. So how these ionic bonds are formed? Because there are R groups in the amino acid structure. So those R groups may be ionizable. So these R groups are ionized under specific conditions or which depends on the surrounding pH. And if the pH is altered, the R group may get protonated or deprotonated upon the changes in the pH, surrounding pH, and so the ionic bonds may break. Hydrogen bonds, similarly, hydrogen bonds are weak. All the non-covalent interactions for that matter are relatively weak as compared to the covalent interactions. And there are major driving force of a polypeptide into three-dimensional conformation are the number of non-covalent interactions. So these non-covalent interactions, because they are weak in nature, they break and the protein structure gets lost. The three-dimensional structure is lost, and that is called denaturation. So how the denaturation occurs? So the denaturation occurs depending on various surrounding conditions, and you must keep in mind that denaturation generally does not involve the breaking of peptide bonds. What are peptide bonds? They are linking the amino acid residues. So apart from the peptide bonds, various other bonds like hydrogen bonds, ionic bonds, these bonds get damaged and the structure is lost. However, generally the peptide bonds, they remain intact. Under extreme conditions, the peptide bonds also break. But in general, denaturation involves the breaking of only these covalent or non-covalent interactions, not the peptide bonds. However, you must also keep the important fact in mind that denaturation leads to the loss of protein activity, or the protein loses its function on denaturation. So we'll discuss some of the factors or some of the conditions which lead to denaturation of the proteins one by one. So the first is strong acids or bases. So how these strong acids and bases, okay, so before discussing on the factors or agents, denaturing agents, the other thing is, depending upon the extent of denaturation, or depending on the extent up to which the three-dimensional conformation is lost, the activity is the function, or the activity or the function is lost. So depending on the extent of denaturation, the protein may partially or completely lose its function. Okay, so if the protein is partially denatured, means it has not completely lost its three-dimensional conformation, so it may retain some part of its activity. However, if the denaturation is to the extent where most of the bonds, most of the interactions have been damaged, then the protein completely loses its function or its activity. Okay, now coming to the agents, denaturing agents, the first is strong acids or bases. So how these strong acids or bases leads to denaturation? Because of the presence of these strong acids or bases, there will be change in pH. Okay, change in pH will further lead to protonation or deprotonation of the R groups, or the side chains of the amino acids, and which ultimately will lead to damaging of the hydrogen bonds, or ionic interactions, or van der Waals interactions, or hydrophobic interactions as well. So because of that, all those factors, because of this protonation or deprotonation, these interactions will get damaged and the protein will lose its three-dimensional conformation, and hence the structure and the function. Now coming to the second point, that is heat or temperature. So change in temperature or an increase in temperature will lead to damaging of the hydrogen bonds, and major hydrogen bonds are very important in compacting this polypeptide into its three-dimensional conformation. So temperature will lead to denaturation of the protein by damaging the non-covalent or weak interactions, and hence the activity is lost. So next is third point, that is heavy metal ions. So how these heavy metal ions, like for example lead and mercury, they affect protein structure because they form ionic bonds with negatively charged R groups, and they also have the capability to bind with the sulfhydryl groups, and hence they they inhibit the normal interactions among the amino acid residues, which causes the folding, and hence the activity is lost because they are interfering in the normal protein, they are interfering in the normal bond formation, they are creating hindrance in the normal bond formation, and hence these lead to denaturation of the protein, and hence its activity again is lost. Then next are organic solvents. So what are organic solvents? Organic solvents, for example ethanol, they also have an OH group because it has an ol, so the functional group is OH here. So this OH group again has the capability to form intermolecular hydrogen bonds, and then they interfere with the normal intramolecular hydrogen bonding, and hence the interfere with the normal hydrogen bonding which occurs between the amino acid residues, and the structure is lost. So next, fifth is detergents. So what do the detergents do? They are amphipathic. Detergents are amphipathic molecules which disturb the hydrophobic interactions because they are amphipathic in nature. For example, chaps. So these detergents, they are amphipathic in nature, and they break hydrophobic interactions which are involved in the protein folding. Next, important is reducing agents. For example, beta-mercaptoethanol. Beta-mercaptoethanol, these reducing agents, what do? They reduce the disulfide bridges or the SS bond into and expose the SH group. So they are reducing this disulfide bonds into and exposing the sulfhydryl group. So SH group again, and because of this the disulfide bridges are broken, so they are essential in forming the tertiary structure. They are in one of the most important bonds. Disulfide bridges are one of the most important bonds which are involved in stabilizing the tertiary structure. So the tertiary structure is lost because of these reducing agents. So these are some of the 6 denaturing conditions which affect the protein structure, and hence these cause denaturation, or you can say they cause the loss of protein activity. So because of these conditions, the protein may lose its entire activity or partial activity. So these are some of the important denaturing agents you must keep in mind. And in the next lecture we'll discuss about classical Anfinsen experiment, where he demonstrated how protein folding occurs, and the correct protein folding is absolutely essential. The correct three-dimensional structure of the linear polypeptide chain, how much it is essential for the accurate functioning. So how accurate it is, how much accuracy it is required for the polypeptide to attain three-dimensional conformation and the function. So how the structure and function are correlated was first demonstrated by Finson, and we'll discuss this his classical experiment in great details in the upcoming video. So till then, stay tuned and thank you.